For our project, we worked on figuring out the optimal growth environment for F. ciliaris under mixotrophic growth based on fucoxanthin and biomass production. Fossil fuels have become a highly demanded material within our society. At least 96% forms of transportation still use petroleum and release high amounts of CO2. Therefore, alternative methods have been looked into as a replacement. For example, microalgae such as diatoms. These single-celled algae, although are resourceful and renewable, still have issues. For example, they're not cost efficient in comparison to fossil fuels. Therefore, within our study, we attempted to reduce the cost by figuring out how to yield the greatest amount of biomass and fucoxanthin concentration. It is believed that F. ciliaris will produce its highest amount of biomass and fucoxanthin concentration when supplemented and under low light intensities, based on former studies shown within Ixia and Garcia's studies. The research question attempted to be answered is, what are the optimal growth conditions of Phytol ciliaris for yielding its high mass biomass and fucoxanthin concentration?
The methods for this experiment were 1,000 microliters samples of diatoms were placed into 6 test tubes. 3 milliliters of F2 and F2 plus glycerol media were then placed into test tubes A, B, C, D, E, and F using a serological pipette. The mixed solutions were then left to grow under various light intensities such as low, medium, high, and high light intensity. 3 milliliter samples of F2 media labeled A, B, and C and F2 plus glycerol media, D, E, and F were placed into vials containing 1,000 microliters of diatoms using a serological pipette. The measure weight of the filter papers were recorded. Specimens were filtered and dried. Filters were weighed, allowing for the biomass to be determined. F2 and F2 plus glycerol filters were trimmed into tiny squares and put into separate tubes. Exactly 4 milliliters of ethanol was assorted into tubes. At a wavelength of 445 nanometers of chlorophyll and fucoxanthin were observed, while at an absorbance of 663 nanometers, only chlorophyll was read for. The results of this experiment show that the diatoms grown in the F2 media supplemented with glycerol had absorbed higher levels and were significantly greater than the diatoms grown in the F2 media. As shown in Figure 1, the absorbance for the algae diatoms for 2 types of media, F2 and F2 supplemented with glycerol. Absorbance was measured 3 times, once on day 0, once on day 7, and once on day 14. The results also show that after filtration, the weight of the diatoms grown in the F2 media supplemented with glycerol was higher than the weight of the diatoms grown in the F2 media. This is shown in Figure 3. As well, the fucoxanthin concentration levels for the diatoms grown in the F2 media were higher than the diatoms grown in the F2 supplemented with glycerol media at OD445 and OD663. This is shown in Figure 4. Fucoxanthin concentration of diatoms after 14 days.
The results of this experiment show that the addition of a growth supplement increases the biomass production and fucoxanthin concentrations of F. ciliaris. The results also show that mixotrophic growth conditions do have a benefit when growing the diatoms. 14 days of growth was observed, and what happens after those 14 days was not measured. So errors could have been errors could have happened in multiple places. The readings could have been off due to errors in the spectrophotometer. Limitations could have also occurred because the algae was not grown in its natural setting. It was grown in a lab under artificial conditions. So basically, the biomass readings, fucoxanthin concentrations, and absorbance readings all had a positive net growth for the 14 days with the added glycerol. In the future, other growth supplements should be studied to see if glycerol had the most positive effect. Also, other types of microalgae besides F. ciliaris should be studied to see if they have a greater yield or they're more efficient in diatom production. And also, the diatom should be grown in different conditions or like weather conditions to see if like what temperature it grows best under.